plants genetic resources too. Dear student, in previous lecture we were discussing about plant genetic resources and different kinds of these resources. Now we will continue that lecture and discuss some other kinds of plant genetic resources. The next one is varieties in the cultivation, breeding lines, special genetic stock, and the last one is the wild forms and wild relative. Varieties in cultivation. Varieties in cultivation are the easiest to use in the breeding program, and they form a major part of working collection. They are good source of gene for yield, quality, etc. They can be introduced in a new area and directly released for cultivation. These plants are selected through specific hybridization, plant selection, or mutation to achieve specific characteristic or trait. Breeding lines. These are the lines of population developed in breeding programs, have narrow genetic base, and often contain valuable gene combination. This group nearly contain homozygous line or mutant lines and lines derived from biology programs, including transgenic lines. They are ordinarily maintained as working collection by breeders. Plant breeding is the art and science of changing the traits of plant in order to produce desired characteristics. It has been used to improve the quality of nutrition in products for human and animal. आमतौर पर ब्रीडर अपने प्रोग्राम में बहुत सारी लाइंस डेवलप करते हैं, लेकिन इस स्टॉक में से बहुत कम लाइंस होती हैं जो कि कमर्शियल प्रोडक्ट के लिए यूज़ होती हैं. वाइल्ड form and wild relative. Wild form are wild species from which crop species are directly derived. They are easy to cross with the concerned crop species. The wild relative include all other species which are related to the crop species by descent during their evolution. And the last one, genetic stock. Genetic stock, broadly defined as plants or population generated or selected for genetic studies, represent a unique and growing class of extremely valuable germ plasm which, depending on crop, types of genetic stock and user community, may represent genetic resources of either transient or long-lasting values. Genetic stock are lines that contain various mutant allele, chromosomal rearrangement, or other cytological abnormalities that are useful only for genetic and basic biological experiments. These resources are used for conserve both economic important species just like corn or yeast, and organisms used purely for research purpose such as fruit flies and bread mold. It is also used in mapping the genes. They can preserve stock for many years. They help in conserving endangered species, used for new hybrid products, and used solely for genetic research and teaching. These genetic resources can be divided into three general groups. The first group is cytological stock, the mutant stock, and the germplasm set. Cytological stock contain chromosomes addition, substitution, aneuploidies in mutant such as induced mutants or the insertion mutant or the tearing population, and in germplasm set mapping population, apparent lines, or the reference germplasm. Any genetic stock collection can represent a few lines to tens of thousands of lines, and therefore can potentially offer a challenge as well as burden to gene bank manager, and from the standpoint of storage and maintenance. और दूसरा चैलेंज ये है कि टेक्नोलॉजी के चेंज होने के साथ साथ न्यू जेनेटिक स्टॉक डेवलप होते रहते हैं, जिससे पुराने जेनेटिक स्टॉक ऑब्सोलेट हो जाते हैं. सो दैट्स ऑल अबाउट डिफरेंट काइंड्स of plant genetic resources.
सो फ्रेंड्स, हमारा वीडियो जो है वो हमारी कंप्लीट हो गई है, ठीक है। अब हम चलते हैं अपनी रीडिंग की तरफ, ठीक है। एज़ यूजुअल कुछ ना कुछ समझ आ गया है मुझे, ठीक है। आप लोगों को भी आई होप आ गया होगा। तो हम चलते हैं अपने हैंडआउट्स में और पीपीटीज में, ठीक है। हैंडआउट्स में चलते हैं। सबसे पहले खुल गया हमारे हैंडआउट्स और इसके अंदर हमारे पास है मॉडर्न कल्टीवर्स सॉरी, थर्ड टाइप है उसकी प्लांट जेनेटिक रिसोर्सेज की, ठीक है। ये है मॉडर्न कल्टीवर्स, द करेंटली कल्टिवेटेड yielding varieties are referred to as modern cultivars. Modern cultivars are also known as improved cultivars or advanced cultivars. These varieties have high yield potential and uniformly as compared to obsolete varieties and land races. Modern cultivars constitute a major part of working collections and are extensively used as parents in the breeding programs for further genetic improvement in various characters, ठीक है। Breeding lines, pre-released plants which have been developed by plant breeders for use in modern scientific plant breeding are known as advanced lines, cultures and stocks. They include advanced cultures which are not yet ready for release to farmers. Sometimes advanced breeding lines in stock are not very much productive but constitute a valuable part of gene pool. Okay, constitute valuable part of gene pool for various economic characters. ठीक होगा। Wild forms of cultivated species. Wild relatives are naturally occurring plant species which have common ancestry with crops and can cross with crop species and are referred as wild relatives or wild species. Wild relatives are important resources of resistance to biotic disease and insect and biotic drought, cold, frost, salinity, etc. or stresses, ठीक है। Wild forms of cultivated species are available in many crop plants, ठीक है। Such plants have generally high degree of resistance to biotic and abiotic stress and are utilized in breeding programs for genetic improvement of resistance to biotic and abiotic stress, ठीक है। Mutation breeding is used as when the derived character is not found, ठीक है, in the genetic stocks of cultivated species and their wild relatives, ठीक है। Mutations do not occur in nature as well as can be mutations do occur in nature as well as can be induced through the use of physical and chemical mutagens, ठीक है। The extra variability which is created through induced mutations constitutes important component of gene pool, ठीक होगा। उसके बाद हमारे बोनस पॉइंट यानी कि पीपीटीज आ जाते हैं हमारे पास। हमारे आ जाते हैं यहाँ पर पीपीटीज जेनेटिक प्लांट्स जेनेटिक रिसोर्सेज टू, ठीक है। इसमें हमारी फर्दर टाइप्स जो हैं वो बताई गई, वेराइटीज इन कल्टिवेशन, ब्रीडिंग लाइंस एंड स्पेशल जेनेटिक स्टॉक, वाइल्ड फॉर्म्स एंड वाइल्ड रिलेटिव्स, ठीक है। Varieties in cultivation are the easiest to use in the breeding program and they form a major part of working collections. They are good source of gene for yield, quality, etc. They can be introduced in a new area and directly released for cultivation, ठीक हो गया। Breeding lines, these are lines or populations developed in breeding programs, have narrow genetic base and often contain valuable genetic combination, ठीक है। This group nearly contain homozygous line or mutant lines. This group nearly contains homozygous line or mutant lines, ठीक है। And lines derived from biology programs including transgenic lines. They are ordinarily maintained as working collection by breeders. ठीक है, उसके बाद wild form and wild relatives. Wild forms are from our wild species from which crop species are directly derived. ठीक है, they are easy to cross with the concerned crop species. ठीक है, the wild relatives include all other species which are related to the crop species by descent during their evolution. ठीक हो गया. उसके बाद हमारे पास जेनेटिक स्टॉक्स ब्रॉडली डिफाइंड एज प्लांट्स और populations generated and or selected for genetic studies represent a unique and growing class of extremely valuable germplasm which, depending on crop type of genetic stock and user community, may represent genetic resources of either transient or long lasting value. ठीक है, उसके बाद हमारे पास आता है जेनेटिक स्टॉक. इसके अंदर जेनेटिक स्टॉक्स कैन बी डिवाइडेड इनटू 3 जनरल ग्रुप्स: साइटोलॉजिकल स्टॉक्स, म्यूटेंट स्टॉक्स और germplasm sets. ठीक है, examples of genetic stock collection: the Charles M. Rich Tomato Genetics Resource Center. ठीक है, बार्ले जेनेटिक स्टॉक कलेक्शन, द बार्ले जेनेटिक एंड एन्यूप्लॉइड स्टॉक कलेक्शन. इसके एग्जाम्पल हैं. सो दिस इज नाउ गुडली और ग्रेटली अंडरस्टूड बाय मी. ठीक है, और आई होप आप लोग भी अच्छे से समझ आ गया होगा. ठीक है, अब हम चलते हैं अपने नेक्स्ट टॉपिक की तरफ, जो है कंजर्वेशन ऑफ प्लांट जेनेटिक रिसोर्स 1. ठीक है, इसकी वीडियो को सुन लेते हैं.
Conservation of plant genetic resources. Dear student, in this module we will learn about what is conservation of plant genetic resources, why do we need to conserve plant genetic resources, and how we can conserve these resources. Conservation is the management of nature and Earth's biodiversity with the aim of protecting species, their habitats, and ecosystem from excessive rate of extinction and erosion of biotic interaction. Germplasm or plant genetic resources has to be maintained in such a state that minimize the risk of loss and allow either its direct planting in the field or its preparation of planting with relative case. This is called plant genetic resources conservation or the germplasm conservation. Now we will discuss why do we need to conserve these resources. There are different reasons behind this conservation. The first one is for food security and agrobiodiversity. We strongly need to conserve these plant genetic resources. The next one is biodiversity provide a valuable source of compound to the medical, food and crop protection and industries. So this biodiversity must be conserved for the future use. The next is all the species are important and help keep the ecosystem balanced. Disturbance in status of any species will lead to disturbance of whole ecosystem balance. So we must conserve all these resources. The last is genetic resources need to be conserved so that they may be used in crop research and be used as sources of genes for crop improvement. Here is some steps which are involved in the conservation of these resources. The first step is we must have some plant genetic resource diversity. Next, we will conserve these resources, and finally we use these conserved products for the production of many different products at a commercial level. Here are different steps which are involved in conservation method of these plant genetic resources. The first one is selection of target taxa. Selection of target taxa decide which species need active conservation and which type of genetic resource is appropriate for that species. The next one is project commission. Project commission formulate a clear, concise conservation statement establishing what species, why, and in general terms where the species are to be conserved. Ecogeographic survey or the preliminary survey mission. This facilitates the collection of all the basic information for the planning of effective conservation and surveys the distribution of taxonomic and genetic diversity, ecological requirement, and the reproductive biology of the chosen species over its entire geographic range. The next one about different objective of the conservation. They formulate a clear, concise set of conservation objectives which state the practical steps that must be taken to conserve the species and propose how the conserved diversity is linked to utilization. Field exploration. Field exploration visit competing potential sites indicated as having high level of target species and genetic diversity by ecogeography survey or the preliminary survey to identify specific location where target species and genetic diversity are to be conserved in genetic reserve. The next step is about different strategies which we adopt for this conservation. This involves the designing, management, and monitoring of these genetic resources. Here are two main strategies about this conservation. The first one is in situ conservation, in which we provide protection to these plant genetic resources inside their habitat, and the next one is ex situ conservation, in which we give protection these plant genetic resources outside their habitat. We will discuss both these types in the next lecture in detail. The next step is conserved product deposition and dissemination. The main conserved product, the plant population of the target taxa, are held in the reserve. However, there is need for safety duplication, and sample of germ plasm should also be periodically sampled and deposited in appropriate reserve with appropriate passport data. After conservation, there is need for systematic evaluation of these conserved products in order to know its various morphological, physiological, and developmental characters, including some special features. Next steps are about utilization of these conserved products at commercial level and obtaining different products due to their utilization. There are different steps which are involved in the model of genetic conservation and utilization, and all these are those steps which we have discussed in the previous slide. Thank you.
So friends, हमारी जो है वो वीडियो वो कंप्लीट हो चुकी है और अब हम चलते हैं आगे। हमारा ये 6 टॉपिक है और इसकी अब हम रीडिंग की तरफ चलते हैं। ठीक है, हम चलेंगे इसकी रीडिंग की तरफ। पीपीटीज और इसके हैंडआउट्स डाउनलोड कर लेंगे और इसकी रीडिंग कर लेंगे। आप लोग देख सकते हैं 3 बज गए हैं, 3:27 हो गए। ठीक है, कर लेते हैं फिर फटाफट से। हमारे टॉपिक्स भी मतलब कम ही रह गए हैं। ये देखिए, अब हम फटाफट से इसको जो है ना रीड आउट कर लेंगे हैंडआउट्स का पहले। ठीक है, तो हमारे पास आ गया है कंजर्वेशन ऑफ, सबसे पहले टॉपिक है कंजर्वेशन ऑफ जर्म प्लाज्म। ठीक है, स्पीशीज और population of a sample of a particular part of its genetic variation can be maintained through in conversation। अच्छा, can be maintained through is conversation। ठीक है, conversation नहीं है, conservation है। ठीक है, A species or a population sample of a particular part of its genetic variation can be maintained through is conservation. ठीक है. The sum total of all genes present in a crop and its various species constitute its germplasm. It is ordinarily represented by a collection of various strains and species. Germplasm provides the raw materials which are equal to genes. The breeder uses these to develop commercial crop varieties. Therefore, germplasm is the basic indispensable ingredient of all breeding programs. ठीक है. Thus, a great emphasis is placed on collection, evaluation, and conservation of germplasm. Conventionally, germplasm is conserved as seeds stored at ambient temperature, low temperature or ultra low temperature. ठीक है. Applications or significance of germplasm conservation. The conservation of germplasm involves the preservation of the genetic diversity of a particular plant or genetic stock. ठीक है. It can be used at any time in future. ठीक है. It is important to conserve the endangered plant as some of the valuable genetic traits present in the existing and primitive plants will be lost. ठीक है. ओके. उसके बाद में मेन क्रॉप्स प्रोड्यूस recalcitrant or short-lived seeds. Similarly, in case of clonal crops, seeds are not the best material to conserve due to their genetic heterogeneity and unknown worth. Their genes need to be conserved. The roots and tubers lose viability rapidly. Their storage requires large space, low temperature, and is expensive. In addition, materials modified by genetic engineering may sometimes be unstable. ठीक है. उसके बाद आ सकता है आएगा मेरे पास such materials are needed to be conserved intact for the future use. ठीक है. Methods of germplasm conservation. The very objective of germplasm conservation or storage is to preserve the genetic diversity of a particular plant or genetic stock for its use at any time in future. In recent years, many new plant species with desired and improved characteristics have started replacing the primitive and conventionally used agricultural plants. It is important to conserve the endangered plants or else some of the valuable genetic traits present in the primitive plants may be lost. ठीक है. A global body, namely International Board of Plant Genetic Resources (IBPGR), has been established for germplasm conservation. ठीक है. Its main objective is to provide necessary support for collection, conservation and utilization of plasma genetic resources. ठीक है. Throughout the world. ठीक हो गया. इसको कर देते हैं क्रॉस. सही है. अब हमारे पास आ जाएंगे हमारे PPTs, हमारे बोनस पॉइंट. ठीक है. इसको हम पढ़ लेते हैं. अब हम इसको फौरन से पढ़ लेते हैं. ठीक है. कंजर्वेशन ऑफ प्लांट जेनेटिक रिसोर्सेज 1. इसमें आ गया व्हाट इज़ कंजर्वेशन ऑफ प्लांट जेनेटिक रिसोर्सेज, व्हाई डू वी नीड टू कंजर्व प्लांट जेनेटिक रिसोर्सेज, डिफरेंट स्टेप्स इन्वॉल्व इन कंजर्वेशन ऑफ प्लांट जेनेटिक रिसोर्सेज. ठीक है. germplasm और plant genetic resources has to be maintained in such a state that minimize the risk of loss and allows either is direct planting in the field or its preparation of planting with relative case. This is called plant genetic resources. ठीक है. उसके बाद हमारे पास कंजर्वेशन और germplasm conservation. ठीक है. अच्छा. कंजर्वेशन ऑफ प्लांट जेनेटिक resources is necessary for. Why do we need to conserve plant genetic resources? It is necessary for food security and agrobiodiversity. Biodiversity provides a valuable source of compounds to the medical, food and crop protection industries. Maintenance of ecosystem. Genetic resources need to be conserved so that they may be used in crop research and be used as sources of genes for crop improvement. ठीक हो गया. Conservation. The link between genetic resources and utilization. Plant genetic resources diversity. फिर conservation और utilization immediate or potential. ठीक है. Utilization produces new varieties New crops, pharmaceutical uses, pure and utilization products है ये, ठीक है. New varieties, new crops, pharmaceutical uses, pure and applied research on farm diversity and aesthetic pleasure etc. इसके बाद हम चल लेते हैं आगे. कंजर्वेशन ऑफ प्लांट एंड aquatic resources, सिलेक्शन ऑफ टारगेट टेक्सा प्रोजेक्ट कमीशन, इको जियोग्राफिक सर्वे और प्रिलिमिनरी सर्वे मिशन, कंजर्वेशन ऑब्जेक्टिव्स, फील्ड एक्सप्लोरेशन, फाइल्ड एक्सप्लोरेशन और उसके बाद कंजर्वेशन स्ट्रेटजीज, कंजर्व्ड प्रोडक्ट डिपोजिशन एंड डिससेमिनेशन, कैरेक्टराइजेशन और एवेल्युएशन, पीजीआर प्लांट जेनेटिक रिसोर्सेज यूटिलाइजेशन प्रोडक्ट्स, ठीक है. मॉडल ऑफ जेनेटिक कंजर्वेशन एंड यूटिलाइजेशन ये है जनाब सेटअप एंड सर्वे, ये जनाब सिलेक्शन ऑफ टारगेट डाटा, प्रोजेक्ट कमीशन, इको जियोग्राफिक सर्वे, उसको कंजर्वेशन ऑब्जेक्टिव, फाइल्ड एक्सप्लोरेशन, कंजर्वेशन स्ट्रेटजीज़, ये वाली दो स्ट्रेटजीज़ हैं, ठीक है. इसके सीड इन विट्रो स्टोरेज, पोलन डीएनए, फील्ड और बोटानिकल जेनेटिक ऑन फार्म होम गार्डन्स, तो ये सब है, ठीक है. कंजर्वेशन प्रोडक्ट्स आगे, फिर उसके बाद कंजर्वेशन प्रोडक्ट डिपोजिशन एंड डिसेमिनेशन, करेक्टराइजेशन और एवेल्युएशन, प्लांट जेनेटिक रिसोर्स यूटिलाइजेशन, फिर यूटिलाइजेशन प्रोडक्ट्स, ये जीन पूल, फिर कंजर्वेशन, फिर यूटिलाइजेशन, सही होगा. इसके बाद हमारा ये टॉपिक कंप्लीट हो गया. आई हैव अंडरस्टूड इट कम्प्लीटली. आई होप आप लोग को भी अच्छे समझ में आ गया होगा, ठीक है. अब हम चलते हैं अपने नेक्स्ट टॉपिक की तरफ. टॉपिक नंबर 7 यह है कंजर्वेशन ऑफ प्लांट जेनेटिक रिसोर्सेज, ठीक है. चलिए इसकी पीपीटीज और हैंडआउट्स पहले डाउनलोड कर लेते हैं, उसके बाद हम इसकी वीडियो देख लेते हैं.
Conservation of plant genetic resources. Dear student, in this module we will learn about different methods for the conservation of plant genetic resources. The method of conservation has been divided into two main types. The first one is in situ conservation and next is ex situ conservation. In situ conservation, as its name is indicating that it is in-site conservation, or we can say that it is on-site conservation. In this type of conservation, we provide protection to animal or plant species inside their natural habitat. This method involves protection of endangered species in their natural habitat. It helps in recovering population in the surrounding where they have developed their distinct feature. This is achieved by protecting this area from human interference. Examples of such areas in which plants are protected in situ conservation are national parks, biosphere reserves, and the gene sanctuaries. These areas are also known as protected areas. Here are some most common types of in situ conservation. The first one is national park, the biosphere reserves, and gene sanctuaries. नेशनल पार्क इन सिटू कंजर्वेशन की एक टाइप है. ये एक ऐसा एरिया होता है जिसको स्टेट की तरफ से वाइल्ड लाइफ की प्रोटेक्शन और प्रोपेगेशन के लिए डिक्लेयर किया जाता है. इसमें वाइल्ड लाइफ को उनका नेचुरल हैबिटेट प्रोवाइड किया जाता है. नेशनल पार्क को इन वाइल्ड लाइफ की प्रोटेक्शन के साथ साथ साइंटिफिक एजुकेशन और तफरीह मकसद के लिए भी इस्तेमाल करते हैं. डिफरेंट एक्टिविटीज़ लाइक forestry, grazing or cultivation are not allowed in these areas. Pakistan has 29 protected areas known as national park. 22 of these are under supervision of respective provisional government and remaining are in private care. For example, Lal Swanra National Park of Bahawalpur. The next one is biosphere reserves. The term biosphere refers to all the land, water, and atmosphere that supply life on Earth. The word reserve means that it is a special area recognized for balancing conservation with sustainable use. Each biosphere reserve demonstrates practical approaches to balancing conservation and human use of an area. Multiple use of such land is permitted. Gene sanctuaries. The genetic diversity is sometimes conserved under natural habitat. In other words, areas of great genetic diversity are protected from human interference. These protected areas in natural habitat are referred as gene sanctuaries. Human activities are permitted to such areas but to a limited level. This type of area has two main advantages. The first one is it protects the loss of genetic diversity caused by human intervention, and second, it allows natural selection and evolution to operate. The next type of conservation, that is ex situ conservation. Ex situ conservation, it is off-site conservation. It means that in this type of conservation, we give protection to animal or the plant species outside their natural habitat. इसमें हम एनिमल्स को या डिफरेंट प्लांट्स को उनके नेचुरल हैबिटेट से बाहर प्रोटेक्शन प्रोवाइड करते हैं और कंजर्व करते हैं, लेकिन वो एरिया जहाँ पर इनको प्रोटेक्शन दी जाती है वो एरिया इनके नेचुरल हैबिटेट से बहुत ज़्यादा रिजेम्बलेंस शो करता है। और इस तरह के प्रोटेक्टेड एरियाज में सीड जीन बैंक्स, बोटेनिकल गार्डन एंड zoological पार्क्स और zoological gardens शामिल हैं। Here are some situations or some condition in which ex situ conservation techniques are implemented. First of all, to safeguard population or individuals that are in danger of physical destruction when protection in situ is not possible. The next is to safeguard population which are in danger of genetic deterioration, to ensure a readily available continuous supply of reproductive material, either creating a production source or through storage. And the last one, it allows commercial improvement of a species through breeding activities and supply of genetically improved reproductive material. Here are some common types of ex situ conservation. The first one is gene bank and then botanical garden. Gene banks. Gene bank refer to a place or organization where germplasm can be conserved in living state. Gene banks are also known as germplasm bank. We have divided seed banks into two main types. The first one is seed gene bank and the next is field gene bank. Seed gene bank. Seed gene bank is a place where germplasm is conserved in the form of seed is called seed gene bank. Seeds are very convenient for storage because they occupy smaller space than the whole plants. In seed gene banks, there are three types of conservation. First is short-term conservation, second modern term conservation, and the third is long-term conservation. Base collection are stored for long-term conservation for near about 50 years or more at 18 to 20 degrees centigrade, and active collection are stored for short-term conservation for near about 3 to 5 years at 5 to 10 degrees centigrade. This type of conservation has some advantages like a large number of germplasm samples or entire viability can be conserved in a very smaller space. In seed gene banks, handling of germplasm is much easy and the germplasm are conserved under pathogen and insect-free environment. This type of conservation has some limitation also. First is failure of power supply may lead to loss of viability and thereby loss of germplasm, and it requires periodical evaluation of seed viability. After some time, multiplication is essential to get new or fresh seed for storage. The next type is field gene bank. Field gene banks, also called plant gene banks, are areas of land in which germplasm collection of growing plants are assembled. In field gene bank, germplasm is maintained in the form of plant as a permanent living collection. They are often established to maintain working collection of living plant for experimental purpose. This type of conservation has some advantages like it provides opportunities for continuous evaluation for various economic character, and it can be directly utilized in breeding program. This type of conservation has some drawbacks also, like it cannot cover the entire genetic diversity of a species. It can cover only a fraction of the full range of diversity of a species, and the maintenance of germplasm in field gene bank is a costly affair. And the next is botanical garden. A botanical garden or a botanic garden is a garden dedicated to collection, cultivation, and display of wide range of plants labeled with their botanical names, where plants, especially fern, conifers, and flowering plants, are grown and displayed for purpose of research and education. Here are some common examples of botanical garden from Pakistan, like Botanical Garden, Governor House, Lahore Botanical Garden, and the Karachi University Botanical Garden. So dear students, it's all about different type of conservation method for plant genetic resources. Thank you.
ओके फ्रेंड्स, सो हमारे पास ये वीडियो भी कंप्लीट हो गई। अब इसका भी हम जल्दी से जल्दी जो है ना इसका मटेरियल, रीडिंग मटेरियल पढ़ लेते हैं। हमने डाउनलोड पहले ही कर लिया था, तो हम सबसे पहले इसका हैंडआउट्स पढ़ लेते हैं, ठीक है। चलो जी, बिस्मिल्लाहिर्रहमानिर्रहीम, यहाँ पर आ जाते हैं हैंडआउट्स पर। देयर आर टू अप्रोचेज फॉर germplasm conservation of plant genetic materials: in situ conservation, ex situ conservation. In situ conservation means the conservation of germplasm in their natural environment by establishing biosphere reserves, ठीक है, और नेशनल पार्क्स और जीन सेंचुरीज, ठीक है, इज regarded as in situ conservation. This approach is particularly useful for preservation of land plants in the near natural habitat, along with several wild relatives with the genetic diversity. The in situ conservation is considered as a high priority germplasm preservation program, ठीक है। The major limitations of in situ conservation are listed below. The risk of losing germplasm due to environmental hazards, ठीक है। The cost of maintenance of a large number of genotype is very high. Ex situ conservation. The ex situ conservation is the chief method for the preservation of germplasm obtained from cultivated and wild plant materials. The genetic materials in the form of genes or from in vitro cultures, plant cells, tissues, or organs can be preserved as gene banks for long-term storage under suitable conditions. For successful establishment of gene banks, adequate knowledge of genetic structure of plant populations and the techniques involved in sampling, regeneration, maintenance of gene pools are essential. ओके जनाब, डन हो गया, ठीक है। इसके बाद हम चल लेते हैं हमारे पीपीटीज की तरफ, ठीक है। हमारी प्यारी अच्छी वाली बोनस वाली अच्छी पीपीटीज की तरफ चल लेते हैं। हमारे पास आ गई कंजर्वेशन ऑफ प्लांट जेनेटिक रिसोर्सेज 2, ठीक हो गया। इसमें क्या पढ़ेंगे? कंजर्वेशन मेथड, इन सिटू कंजर्वेशन और एक्स सिटू कंजर्वेशन, सही हो गया। कंजर्वेशन मेथड में दो है, इन सिटू कंजर्वेशन, एक्स सिटू कंजर्वेशन। इसमें कंजर्वेशन प्रोडक्ट्स, सीड, लाइव प्लांट्स, इन विट्रो एक्स प्लांट्स एंड डीएनए, पोलन एंड डेटा, ठीक है। इसके बाद हमारे पास आ गया है इन सिटू कंजर्वेशन में, दिस मेथड इन्वॉल्व्स प्रोटेक्शन ऑफ एंडेंजर्ड स्पीशीज इन देयर नेचुरल हैबिटेट्स, ठीक है। It helps in recovering populations in the surroundings where they have developed their distinct features, ठीक है। For examples are national parks, biosphere reserves, gene sanctuaries, ठीक हो गया। इसके बाद हमारे पास सारे types of in situ conservation, national park, biosphere reserves, gene sanctuary, ठीक हो गया। ये national park है। national park may be defined as a area declared by state for the purpose of protecting, propagating wildlife there in or its natural environment for their scientific, educational and recreational value. Here biosphere reserves are areas of terrestrial and coastal ecosystem promoting solutions to reconcile the conservation of biodiversity with its sustainable use. They are internationally recognized, nominated by national governments and remain under sovereign jurisdiction of the states where resemble their natural habitats. ठीक है, so examples are seed gene bank, botanical gardens, zoological parks. ठीक हो गया. उसके बाद हमारे पास है ex situ conservation. ठीक है, ex situ conservation में हमारे पास आ गया. ex situ techniques are implemented in well defined situations to safeguard populations or individuals that are in danger of physical destruction when protection in situ is not possible. ठीक है, to safeguard populations which are in danger of genetic deterioration. सही, to ensure a readily available continuous supply of reproductive material, either creating a production source through storage. ठीक है, to allow a commercial improvement of a species through breeding activities and supply of genetically improved reproductive materials. सही हो गया. इसके बाद हमारे टाइप्स ऑफ ex situ conservation, gene bank, botanical garden. ठीक हो गया. हमारे पास gene bank में आ गया. Gene bank refers to the place or organization where germplasm can be conserved in living state. Gene banks are also known as germplasm banks. ठीक है, इसके बाद हमारे पास आ जाता है, इसके बाद हमारे पास आता है seed gene bank. A place where germplasm is conserved in the form of seeds is called seed gene bank. Seeds are very convenient for storage because they occupy smaller space than whole plants. ठीक है. Field gene bank. Field gene banks, also called plant gene banks, are areas of land in which germplasm collection of growing plants are assembled. ठीक हो गया. इसको botanical garden. Botanical garden or botanic garden is a garden dedicated to the collection, cultivation and display of a wide range of plants labeled with their botanical names. ठीक हो गया. So thanks Lord. ये बहुत अच्छे से टॉपिक समझ में आ गया है. ठीक है, हमारी डेली लाइफ से रिलेटेड था. ठीक है, अब हम यहाँ पर इस वीडियो को एंड करते हैं. ठीक है, बाकी का हम अगली वीडियो में करेंगे.